Hello everyone. My name is Parsa. I finished my master's here at UTMB Galveston in pharmacology and toxicology, and I would like to present one of my projects here in terms of studying these two ion channels. We utilize the porta patch, and so I want to show you guys some of the things the porta patch can do in terms of drug discovery and some of the work that I've done. So my title of my presentation is Intracellular Fibroblast Growth Factor 14 Peptide Differentially Regulates Nav1.2 and Nav1.6 function. So to give start of the presentation, what I wanted to do is I wanted to talk a little bit about how some of the current drugs and molecules that modify and modulate voltage-gated sodium channels work. Most of them bind to extracellular portions of the channel. Some of them bind to the pore, like TTX. Some of them bind to voltage sensors on the outside. And very few of them modulate the interior side of the channel. So there's a space to improve or develop novel therapeutics from that sense. And so here I have a picture of some of the different structures in terms of how drugs interact with various domains of the channel. Here are two really important antiarrhythmics that are known to be used in cardiology, antiarrhythmic 1C and 1A, I believe, and they're used widely. And so the main point of this slide was to kind of let you guys know that there is a lot of space in terms of real estate to be modulating these channels. And in addition, a lot of these peptides and not peptides, a lot of these small molecules interact independently of auxiliary proteins. And so the auxiliary protein that we're going to be talking about is the intracellular fibroblast growth factors. These they lack the exportation signal on the protein, and so they're not thrown out into extracellular space, and so they remain intracellular and have a lot of interesting biological functions. And so from the inside, a lot of these FGFs can be phosphorylated via kinases, and they can be dephosphorylated via phosphatases. We do know that some of these FGFs can create homo and heterodimers with other iFGFs. Some of the other ones include 13 and 14, and I believe 12, 13, 14 are all intracellular. And some of them can even bind to microtubules. They can help stabilize the cell that way. And so what we're doing here is we're trying to understand the biology, the physiology of how these FGFs interact with the channel, characterize that, and see which diseases may even have these iFGFs part of their pathophysiology, and how we can modify or modulate the interface between FGFs and sodium channels. So the FGF comes in, phosphorylated and unphosphorylated versions will bind to the channel, cause changes in the ionic current. And what we're trying to do is we're trying to throw something either in between the auxiliary protein in the channel and prevent them from binding, or we could create new pseudo iFGFs, essentially where a small molecule or peptide can bind to iFGF and cause a conformational change, and that whole complex can interact with the channel, giving you an interesting ionic signature. Or you could have a situation where the peptide simply interacts with the channel irregardless of the iFGF. In this case, that is one of the possibilities that we were trying to explore in this experiment. To give kind of like an overview of what sodium channels are, they're isoforms. So sodium channels come in 9 different flavors from 1.1 to 1.9. And interestingly, they are heterogeneously expressed throughout the human body, meaning that they're not all in the same spot, and they're not all in the same cells. So they have cell specificity as well as location specificity. And these inherent built-in features of these channels can provide selectivity to certain areas of the body. And so We picked the 2 sodium channels in this study that we picked were Nav1.2 and 1.6, and we had interest in the brain, the central nervous system, and so these are abundantly expressed. And one of the interestingly, one of the example cells that we wanted to focus on were pyramidal cells. And they have a very interesting biological configuration of these 2 channels at the axon initial segment, which is the portion where action potentials are generated. And so biologically, these channels are put together in a very interesting layout where you have more 1.6. This isn't a mature cell because this would be different in developmental stages. So these channels change over time. But in a mature pyramidal cell, you have 1.6 more at the distal axon initial segment here, and you have more 1.2 relatively towards the proximal axon initial segment and more into the soma, into the dendrites. So from a perspective of understanding how action potentials propagate inside the cell, 1.6 is mainly responsible for propagating forward action potentials, while 1.2 is mainly responsible in coordination with other voltage-gated sodium channel, in coordination with other voltage-gated channels like potassium channels, helps propagate back propagation into the dendrites. And so we wanted to see if our drugs that are modulating iFGF and sodium channels, if they have any ability to modulate the channel independently of iFGF. So we talked about the isoforms. We talked about how they're located inside the cell. And so getting even more granular, we'll talk about a little bit some of the mechanisms of voltage-gated sodium channels. So these channels have been studied for quite a long time, and they have various conformations, meaning they're not always conductive, and they have certain properties as how they conduct the flow of sodium channels through them. So at rest, at negative 80 millivolts, for example, the channel will be in a closed state. The voltage sensors within the domains 1 through 4 are situated in the plasma membrane and are not going to be sticking out because the voltage difference across the membrane isn't sufficient enough to drive the positive lysine charges that exist on the S4 segment outwards. As you slowly start depolarizing the cell, eventually you'll open up the channel, and activation is fast, and what we call this steady state activation. And as the channel is open quickly, as those S4 segments move out, an inactivation particle has access to its binding partners and immediately will inactivate the channel. But the channel can also exist, interestingly, in a closed inactivated state, meaning that its subthreshold depolarization is not sufficient enough to move the S4 segments out of the way. If, for example, maybe 2 S4 segments from 3 and 4, domains 3 and 4, move out, it isn't enough to open the channel, but it is enough to allow the binding partners of the inactivation particle to access, and therefore you can inactivate the channel without having opened it. And so there's a lot of interesting dynamics with this channel, and this is just speaking of the channel alone. We haven't talked about how iFGF can come in and modulate these. So the goal of this whole project was to identify compounds or peptides, in this case, that prevent the interaction between iFGF and sodium channel. So in A, we have a screening in which we've transfected hex cells with 2 different types of plasmids. One plasmid containing a CD4 segment, which essentially puts the so CD4 connected to the C-terminal domain, which is the portion of the sodium channel which we believe iFGFs are interacting with. We essentially put that into the cell, and now you have not the whole channel, but a portion of the channel sticking out into the cytosolic space. And then we have another plasmid containing the iFGF14 that has split luciferase attached to it. So one portion of the luciferase enzyme is on one protein and the other is on the other. And in the split luciferase complementation assay, if these 2 come together, you'll see luminescence. If they are disrupted, the luminescence will drop. And so we're testing these peptides that are derived from the interaction between iFGF and Nav1.6. So this valine we believe to be really important for that interaction. And so what we've done is we created a peptide scaffold called FLPK. And then out of that scaffold, we've created peptide derivatives, peptidomimetics, essentially, from FLPK. And we've created many of them, here's 20, and we've tested them in a split luciferase assay. And what we found is number 13, which is ZL017, has a really great C-log p-value, as well as a great ability to negatively control complex assembly. And so what we've done is we did a counter screen against the full luciferase enzyme. This would support the idea that these compounds are not interacting with, let's say, a catalytic site on the luciferase enzyme, and that in fact these peptides are preventing complex assembly. And we have cytotoxicity studies here, and we've done a dose response to calculate an IC50 value. Here. So what we've done is we've decided to do a selectivity study with this molecule to see if its mechanism of action is required, if its mechanism of action requires FGF to be involved, because that's where it was originally found. And we found that this peptidomimetic actually has intrinsically is able to bind to the alpha subunit alone and independently of IFGF, and it can have some effects. We're noticing that it has the ability to modulate peak current density. You see the IV curve here, in which we're giving, these are the two voltage protocols that we applied. The first voltage protocol gives us the IV curve and the peak current density and activation kinetics here. So this is steady state activation. And what we see is with 10 micromolar of this drug is we're seeing a hyperpolarizing shift in the V half of activation of this drug, meaning that this channel with this drug opens a little bit sooner in the action potential cycle, which is quite interesting. Continuing on, we also studied the inactivation kinetics, as mentioned earlier. So inactivation, this is steady state inactivation, in which the inactivation particle binds to its binding partners after the channel has been in the open state. And we're also seeing a hyperpolarizing shift as well. And so we have a very interesting phenomenon that we're seeing with this peptide. And in addition, we studied how this peptidomimetic interacts with 1.6. Again, we're seeing a very different type of layout here. We are seeing changes in peak current density, but we're seeing a depolarized shift in voltage of activation, steady state activation. And in terms of inactivation, we're seeing, again, the same sort of depolarizing shift. And so what we tried to do is we tried to rationalize these effects by looking at the mechanism of action of how currently sodium channels are thought to work, and kind of backtracking maybe where is this peptidomimetic binding and how is it causing its effects on the channel. And so we highlighted some spots that maybe we think are important to causing those effects, especially the S4 to S5 loops on the intracellular side, as those are connected to the voltage sensors. And if anything is pulling or pushing on these loops, they would essentially give you the signature of either hyperpolarizing or depolarizing activation. And we also have marcations on the inactivation gate. And based on the literature, the first 100 amino acids that are coming out of the C-terminal domain are thought to be more important than the distal amino acids in inactivation. So for 1.2, we're seeing inactivation effects. For 1.6, we're not. So we're trying to rationalize that. And so we came up with a hypothesis as to where things are binding. And then we decided to test those in silico by doing by using AlphaFold. So currently there are no full structures, cryo-EM or crystal structures of the full sodium channel of 1.2, 1.6. So what we decided to do is we decided to use AlphaFold, which has never been done before, to render these channels and superimpose the structures onto segments that are crystallized. So there is cryo-EM of certain parts of the channel, but not the whole channel. So we decided to create a model here, and we decided to do docking studies, and we found that for 1.2, ZL has 3 hydrogen bonds to the inactivation gate, and has 1 hydrogen bond to the proximal C tail. And so this kind of, in relationship to 1.6, we're not seeing those strong hydrogen bonds with the inactivation gate, a very weak pi interaction here. And again, a hydrogen bond at the distal C tail. So from these results, we're happy to see these results, as they support our EFIS data and saying that the ligand has a stronger effect on inactivation gate, meaning that accounts for the effects of ZL on inactivation for 1.2 and not for 1.6. So to kind of wrap up my talk, so these channels are situated at the axon initial segment, and what this peptidomimetic is essentially doing is it's reducing the peak current density coming into the cell, and this in hyperpolarizing 1.2 and depolarizing, shifting in a depolarizing manner of 1.6. This would essentially make these 1.2 channels open up sooner and make the 1.6 open up later. So this would have a very interesting effect in terms of how it would influence action potential kinetics. And we do have some in silico modeling of this, which I don't have shown here, but it will be in the paper because this is part of a manuscript that we are trying to prepare. And it'd be interesting to see not only the acute effects, but especially the chronic effects of this drug in terms of how the receptor profile of these channels would change over time. Maybe the cell will pull some 1.6 off the surface and replace them with another isoform, given the application of this drug over a long period of time. And so I just want to thank everyone in my lab that helped with these experiments. And I'm a new electrophysiologist. I've only been doing this for 2 years. And so the porta patch has been really, really beneficial in my learning, as I don't have the high level skill of an expert electrophysiologist. So the porta patch was very easy to use and very useful in conducting these studies. So I'd like to thank everyone for coming, and if there's any questions, I'd be happy to take them.
Thanks a lot, Parsa. Very interesting talk, and very good or nice for us to hear that you like the porta patch so much, because it's so easy to use and it contributed to your research. We actually have some questions here in the chat. Rod from our ES team was asking, how did you add the compound? Do you use a perfusion system, or do you pre-incubate your cells before you do the recordings, or?
Pre-incubation, yes. We usually pre-incubate for around 30 to 45 minutes.
Another question we have in the chat: have you tried the effect of the protein on other channels located on the axon segment, like potassium channels?
No, which would be very interesting to do, because we do know that FGF14 does interact with some potassium channels, and so given that this peptide was derived from the interaction of the intracellular fibroblast growth factor and the sodium channel, there may be some overlap in the ability of this peptide to bind to other voltage-gated sodium channels. And in addition, this peptide may even have multiple binding sites on the isoforms that we were talking about, because when we were doing our AlphaFold modeling, we had multiple high probability sites that came up in the docking, and so what we did was we selected just the intracellular ones, but you know, who's to say that there aren't multiple binding sites for these drugs? And I think personally, that's good for drugs. If you try to be too selective with drugs, sometimes you lose efficacy that you're trying to have, especially when you get to patients and get to people and stuff like that outside of the
lab. Here's another question: is there a tissue specificity for different IFGFs?
Yes. I know that DRGs, not all DRGs so in DRGs there are 1.7 is kind of like the one that everyone's talking about, 1.82. But some DRGs express FGF13 without 1.7. But every time 1.7 is expressed in a DRG, I believe FGF is also expressed. And so yes, there are some cells that it can express FGF13, some that do not. But out of the exhaustive list, out of the entire body, I don't have them all memorized. But I know the nucleus accumbens, which is what we study in this lab, that is really a brain region that we're really interested in, does express FGF13. And in addition to that question, not all FGFs have the same effect in different cells. So you could be in one cell type, you could see maybe a reduction in peak current density, maybe a depolarizing shift. Another cell, you get a different effect. Maybe it reduces action potential propagation. In one cell, it increases it. Because these FGFs are not alone, there are other proteins that can bind to these channels. You kind of need to be thinking of FGF as kind of like one player in a massive complex. Some kinases like GSK3 beta can bind to these sodium channels and act as a scaffold protein. Another interesting protein that we've been studying in the lab that came out of one of my friends. He's studying creatine kinase B. So that's a very interesting protein, because we in the lab we kind of have this idea that metabolism is heavily linked to the expression of and modulation of these channels. So we found a creatine kinase B to also. So again, all these proteins, different disease states, different quantities come together and they can modulate these channels.
There comes an add-up to that question with the tissue specificity. What effect would the peptides have when administered systemically in vivo? Is there something you can say about that?
So I know a lot of people don't like peptides because they can degrade very easily. They're not very stable in the plasma. I don't have those studies, but I think we've done those studies. And again, this is not a true sense of the peptide, is a peptide derivative. So we took with peptide and we added different moieties to it. So, but who knows? Maybe drug delivery in the future gets better. The drug delivery technology, and we're able to encapsulate these peptidomimetics in some sort of vesicle that can give us even more specificity, and we don't need the fact that the cells are hard to get to. We can, maybe use an exosome. I don't know. We put certain proteins on the outside of the exosome. We can kind of send this, package it to a certain type of cell. But from the classical sense, yeah, I see what you're saying. Yeah, there's definitely an issue with plasma stability for sure.
And one last question before we're running out of time. Did you have a look at use-dependent effects of those peptides on the channels?
Like long-term inactivation, we did not run those studies, no. But I should have. I have the protocol, but we didn't show that information, no.
Okay, thanks. Then thanks again for your great talk, and we'll curious to hear another one from Elizabeth.
Thank you.